All right, so chapter 3 deals with the basic principles of heredity. Okay, so this chapter is really going to focus on the principles of heredity and how genes such as hair color, eye color are passed on from one generation to the next, and how certain factors like dominance influence inheritance. So this diagram right here is a picture of two children from the Solomon Islands, which were first inhabited about 30,000 years ago, when the Neanderthals were still in northern Europe. What's interesting is that in the Solomon Islands, most of the people there have dark or more melanated skin, but they have about 5 to 10% have blonde hair, which is the highest frequency of blonde hair outside of Europe. So you may be wondering, how did the Solomon Islanders get their blonde hair? Okay, there have been so many hypotheses and theories about how this occurred, from mutations to bleaching from the sun and salt water. And it was still a mystery until about 2012, when geneticists went to the Solomon Islands and began to do studies on them. And what they did was they collected saliva and hair samples from over 1,200 people on the island, and they extracted the DNA from those samples. They were able to determine that there was a strong correlation between the presence of blonde hair and a specific genetic variant, this gene located on the chromosome 19, which was the TRP1 gene, which stands for tyrosinase-related protein 1. And that specific gene encodes an enzyme that plays a role in the production of melanin and its known behavior or ability to affect pigmentation in mice. They found that specifically there was a single base difference between the DNA of the islanders with the blonde hair and that of islanders with dark hair. The blondes had a thymine base instead of a cytosine in their TRP1 gene. They further found information that allowed them to identify that it was a recessive trait, meaning that both parents had to give the recessive allele to the offspring in order for them to get blonde hair. So ultimately, dark hair is dominant or more prominent than that of blonde hair. Okay. So this is what we're going to go into, and understanding in this chapter is how are traits inherited and what factors play a role in that. This brings us to Gregor Mendel. Gregor Mendel was a priest in Vienna, and he did a lot of his work because at this monastery that he was staying at had a nice garden. He was able to really get into agriculture there, and he began to, after his studies and getting his training, he began to study the pea plants, and he conducted breeding experiments from about 1856 to about 1863, and he actually published his work. Now, there was much significance to his work where he looked at, used these pea plants to study different attributes and characteristics of these pea plants and how they were passed on from one generation to the next. But what was interesting is a lot of his work that he did not get recognized until the 1900s, when 3 botanists picked up his work and they did independent studies, and they found similar results to what Mendel did. His work was very effective for several reasons. One is his choice of a model system. He used pea plants. Now, pea plants, typically, they don't have a short lifespan like you would expect in fruit flies, which are currently used in genetics. But given Mendel's time and his ability, it was about a year to collect the data. But that was sufficient for him, given what he had access to. Also, another good model system would have been bacteria, as we learned in chapter 1. However, he was able to conclude much synopsis and conclusions from his work, and it was very beneficial in helping lay the groundwork for what we know about genetics today. With the pea plants, he studied 7 different traits, as you see in this diagram here, such as seed color, shape, seed coat, the pod color, pod shape. So he measured and looked at various traits, and he applied mathematics to help him understand his work, which was really unique at that time because he was one of the first to do that. And he also looked at patterns from his results to see how these patterns were playing a role in the inheritance of these traits in these pea plants. So in order for us to really dive into this, I want to talk about some key terms that are really, really important for you to understand in moving forward in genetics. One is a gene. Okay, a gene is an inherited factor encoded in the DNA. Okay, it's a segment of DNA that helps determine a characteristic. Okay, it helps determine a characteristic. An allele is an alternative form of a gene. Okay, genes are located at a specific locus, meaning a specific place on a chromosome, and the genotype, or the combination of the 2 alleles for a specific trait, make up the genotype of an individual. And when you look at an individual that is what we consider diploid, meaning they have 2 forms or 2 copies of their genetic information, they're considered an heterozygote or heterozygote when they possess 2 different forms of that allele at a specific locus. They're considered homozygote if they have the same, allele at that locus. Now, when you're looking at one's trait, the actual physical manifestation of that trait is called the phenotype. It's the appearance. It's what you see with what you can see with your eye. Okay. And then the characteristic or character refers to the attribute or the feature possessed by a specific organism. So in terms of the visual example of the genes and alleles, you see that there's 2 chromosomes here. Okay. Let me just highlight them for you. You have chromosome 1 here, and then the accompanying chromosome here. I'm just going to mark that as 2. Okay, these are homologous chromosomes, meaning they're the same chromosome, one from the mother. Okay. Excuse my drawing, you guys. It's not that easy to draw with this. And then one from the father. Okay. Now, on this chromosome, at a specific locus or a specific place, there is a gene for a specific trait. In this case, we're going to say that it's gene R. Okay. Gene R for, let's say, the shape of the pea, whether it's round or wrinkled, okay, is located in the same spot. However, one allele or chromosome can have the allele for the outer coating of the pea plant, but it could be that it's going to be, in this case, round, okay, which is denoted by the dominant allele R. Or that same feature on the opposite chromosome, okay, could have the recessive allele for that same trait, and it produces wrinkled seeds, okay? Same trait, different forms of that gene. Okay, so it's important to understand that genes exist in different versions, and those versions of the gene are called alleles. Okay, different alleles for a particular gene occupy the same locus on homologous chromosomes. So in this case here, for this organism, because they have a big R, which I'll write here, and a little r, they're considered heterozygous. They would be considered homozygous if they had 2 big alleles, or 2 dominant alleles, I should say, or 2 recessive alleles. And in this case, we would call them homozygous dominant or homozygous recessive. This case is heterozygous. So let's go a little bit into the work that Mendel did, looking at his pea plants. Now, one of the things that Mendel did, he did what we call, or created, crosses. Crosses allow you to breed, okay, 2 different organisms and look at the outcome. So he started off with 34 varieties of peas and spent 2 years selecting those varieties that he would use in his experiments. Okay. He began studying the monohybrid crosses, mono being one. Okay, so he looked at one single characteristic, one trait, in one experiment. He crossed a pea plant that was a pure breeding, which is homozygous, okay, for round seeds, with one that was pure breeding for wrinkled seeds. So one was homozygous dominant and another one was homozygous recessive. The first generation that was produced is called the parental generation, or the P generation. That's what you see here. This is the parental generation. Okay. Round versus wrinkled. Now keep in mind, he's only going off of the phenotype. Okay. He then took from the P generation, okay, the offspring of the parents in the P generation produced the F1 generation, F standing for filial, okay? And that's what you see here. He allowed them to self-fertilize and then looked at the offspring from the F1 generation, which is now considered the F2 generation. Okay? And he did, for the F1, he did about 60 crosses. He then turned around and did reciprocal crosses, okay? And that's where you allow the male gamete to fertilize the female gamete of different traits, okay? And now what Mendel ended up concluding from his studies of looking at these different pea plants was that one character is encoded by two genetic factors. Okay, and remember at this time he didn't know the terms genes and alleles. So he's calling these alleles factors. His second conclusion was that two genetic factors, or alleles, separate when gametes are formed. His third conclusion was the concept of dominant and recessive traits. And then the fourth conclusion is that two alleles separate with equal probability into the gametes. Now, how did he come to these conclusions? Okay. One, you have to remember that when he crossed this parental or this P generation to produce the F1 generation, all of the F1 seeds were round. Okay. But remember, in the parental, one of the parents was wrinkled. So he did not see the same phenotype show up in the F1 generation. Okay, so this was helping him to come to this conclusion. Then when he did the F2 generation, or produced the F2 generations, he saw something that was different from the F1, that the trait from the P generation actually came back or showed up in the F2 generation, where he had a fourth of them that were wrinkled. So he had a 3-to-1 ratio, okay, of round to wrinkled seeds. And this was the phenotypic ratio. 3/4 were round and 1/4 were wrinkled. Okay. So the traits of the parent plants, they didn't blend. Although the F1 generation displayed the phenotype of only one parent, both traits were passed on to the F2 generation in that 3-to-1 ratio. And this really was really foundational and novel, because it really led to the understanding of this dominance versus recessive. So Mendel's first law is the principle of segregation. And what this suggests is that each individual diploid organism possesses two alleles for any particular characteristic, any trait. These two alleles segregate when the gametes are formed, and one allele goes into each gamete. So ultimately you have the separation of gametes and only one is passed on. Okay. Then you have the concept of dominance, right? When two different alleles are present in a genotype, that will be considered a heterozygote. Only the trait encoded by one of them, the dominant allele in this case, is observed in the phenotype. Okay. Another way of thinking about dominance versus recessive is whatever phenotype shows up more prominent or is more prevalent, that is going to be your dominant trait. And whichever shows the least is going to be your recessive. Okay. Now keep in mind that when it comes to dominance, we can't always denote the genotype of a dominant phenotype, because it could be a carrier, meaning a heterozygote. Now, when it comes to monohybrid crosses, okay, they're explained by the principle of segregation. Okay. That's what we just talked about here. Mendel's first law, the principle of segregation. The symbols in these crosses correspond to the alleles on the chromosome. And we get that from Sutton, the chromosomal theory of inheritance or heredity. So here is a monohybrid cross. Okay, monohybrid cross. And as we saw before in the prior slide, the Mendel's monohybrid cross revealed the principle of segregation in the concept of dominance. And this is how we were able to see the change in the phenotypes that were the traits that were showing up, also known as characteristics, from one generation to the next. We see it in the P generation, but then it disappears in the F1 generation and then reappears in the F2 generation. Okay, and then we see it even more pronounced in the F3 generation. Okay, so this understanding of the principles of segregation is very clear in a monohybrid cross, meaning one characteristic being studied, or one gene being studied in this monohybrid cross. So in terms of the comparison of the principle of segregation and the independent assortment, we can look at some of Mendel's law. So we know his first law that we've already talked about. And if you look and go back to meiosis from chapter 2, okay, everything that we talk about in genetics is connected. Okay, so don't look at it in a compartmentalized manner. Everything is connected. So in terms of Mendel's first law, understanding that each individual organism possesses two alleles that encode a trait. You see this before meiosis. Okay, the alleles separate when gametes are formed. We see this in anaphase when the chromosomes start to separate and go to opposite poles. We also see that the alleles separate in equal proportions. That's also in anaphase 1. And then in Mendel's second law, which is the independent assortment, suggests that alleles at different loci separate independently of each other. This is also seen in anaphase 1. Now we have to consider that, or assume that no crossing over, okay, occurs. And if crossing over takes place, then the segregation and independent assortment may also occur in anaphase 2 of meiosis. So when we talk about segregation, right? Think about the separation of the homologous chromosomes back in meiosis, because this is a very important aspect of it, okay? How does this relate back to meiosis? I want you to think about the chromosomal theory of inheritance, okay, that was developed. The symbols that are used in genetic crosses, like the big R and little r, are what we call shorthand notations for particular sequences in DNA. So you're not going to look at the DNA and see an R, a big R, a little r, but it's just the way that we notate specific genes in the DNA. The two alleles of a genotype are found on different but homologous chromosomes. So in this case here, you see there's one chromosome here, and then there's another chromosome here. They're the same chromosome, but one chromosome that may be derived from one parent is going to have a dominant allele for that trait, and the homologous chromosome derived from the other parent is going to have the recessive allele on that chromosome. Once it undergoes replication in S phase, or DNA replication, it's going to double. This should look familiar. There's our centromere, okay, and you have your sister chromatids. Okay, in prophase 1 of meiosis, crossing over may or may not take place. It is cell dependent. Okay, if you don't have crossing over, then what you have is 4 chromatids. If crossing over does occur, you'll see that there has been a physical exchange of genetic information, and that the chromosomes are not necessarily the same. You'll see this chromosome here is not present on this end over here, on this side, when crossing over did not take place. Also, this same chromosome here is not present on this end. Okay, so ultimately, in anaphase 1, when the homologous chromosomes separate, okay, if no crossing over has taken place, then the two chromatids of each chromosome segregate. In anaphase 2, they're identical. But if crossing over has taken place, the two chromatids are no longer identical, and the different alleles segregate in anaphase 2. So this should show you and reaffirm what we learned about crossing over, that it does provide genetic variation, and the presence of crossing over in meiosis provides for genetically unique offspring. Okay, so when it comes to these monohybrid crosses, they reveal, again, as we already stated, the principle of segregation and the concept of dominance. Now, here what you see is what we call a Punnett square. Okay, a Punnett square, it was developed in 1917 by Reginald C. Punnett, and it was a way to illustrate the crosses that were carried out by Gregor Mendel. Okay. So in here you see, this is one parent, okay, and the genotypes that they have to potentially pass on, and then this is another parent. So what you're going to do is you make a box, okay, you illustrate a grid, and the grid is going to list the gametes that were produced by each parent. So in this upper edge, this is one parent, this is the gametes from one parent, and then this is the potential gametes from another parent. Okay, each cell, that's what you see here, 1, 2, 3, and 4. So there's a total of 4 cells in this Punnett square. And each cell is then filled in with one allele from each parent. So in this cell right here, we have this recessive T or lowercase t from the parent. And then this capital T or the dominant T goes in this one here from this parent. So what you have is a heterozygote or a carrier for the recessive T. Okay, and in this case, what we have in terms of our genotypic ratio, we have 1 to 2 to 1. Okay, I'm sorry, this is actually a big T right here. So this is a 1 to 1 ratio. 1 or 50% are carriers, and the other 50% are homozygous recessive. If we look at the phenotypic ratio, it's still a 1 to 1. That is 50% of the offspring, potential offspring, are going to be tall and 50% are going to be short. The reason why these are considered to be tall is because of the law of dominance. As long as one dominant allele is present, it's going to exhibit the dominant phenotype. And in this case, the dominant phenotype is being tall. Here, because there's no dominant allele, then they're going to exhibit the recessive phenotype. This is very important when you're doing crosses or you're testing for potential outcomes, and it allows you to apply a mathematical method to understanding the concept of inheritance. This brings us to probability. And the reason why this brings us to probability, and I should have stated in the first chapter, every class that you're going to take in the sciences can always be tied back to genetics, whether it's chemistry, whether it's math, stats, molecular physiology, anatomy, they all tie back to genetics, because if we're talking about living, if we're talking about life, it has to tie back to genetics. Okay. And so that does not exclude statistics. One aspect of statistics and math is being able to look at probability, which is the likelihood of something occurring or a particular event occurring. Okay. So with probability, it's always going to range between 0 and 1. And it's used in genetics to predict the outcome of many different genetic crosses and even pedigrees, whether a disease is prominent or not. In order to really understand and do calculations for a probability of certain events, we can apply 2 rules. Okay, there are 2 rules of probability that we can use for genetic crosses. The first one is the multiplication rule, which states that the probability of 2 or more independent events taking place together is calculated by multiplying their independent probabilities, or the addition rule, which states that the probability of any 2 mutually exclusive events is calculated by adding the probabilities of the events together. So let's look at the multiplication rule. A great example of the multiplication rule is using dice, okay, or dice. If you were to roll a die, which is a single, okay, one of the dice, In a large number of sample rolls, on average, out of 6 times, you will obtain a 4. Okay, so the probability of obtaining a 4 in any roll is 1 out of 6. Why? Because there are 6 sides to a die. Okay, and if you roll that die again, the probability of getting a 4 again is 1 out of 6. So if you were to look at the probability of getting a 4 on the first and the second roll, you're going to multiply 1 over 6 times 1 over 6. The reason why you're multiplying the probability of those events is because they're independent of each other. Each roll is not depending upon the second roll. So you would apply the multiplication rule. And in that case, it would be 1 over 36. The addition rule is different. Okay, if you were to roll a die for, in this case, on average, 1 out of 6 times, you're going to get a 3. Okay, and 1 out of 6 times, you'll get a 4, depending on your rolling. So that probability of getting either 3 or 4 is 1 over 6 plus 1 over 6, which is equal to 2 over 6, and you have to reduce that down to 1 over 3. Now, these are mutually exclusive events. So this is why we apply the addition rule for looking at this probability. Now, how does this work for crosses? Okay, let's look at, for the example, before we even get into binomial expansion, let's look at applying this to genetic crosses. So let's go back to this case here where we have, let's say, potentially two gametes that have this genotype, big T, little t. Okay, the probability of getting a big T from this parent is 1/2. So we do 1 over 2. The probability of getting a lowercase t is also 1/2. Because these are independent events, we apply the multiplication rule, and as a result, our total probability of getting this genotype is 1 over 4. And as a result, it's going to produce a tall phenotype. Okay, tall phenotype. So this is how we can apply the multiplication and the addition rule to genetic crosses. All right, so let's go to binomial expansion, because when you're doing binomial, you will apply binomial expansion for very complex situations. Okay, so let's say, for an example, you want to know the probability of a couple having 5 children, and out of the 5 children, 2 are going to have albinism, 3 with pigmentation. Okay, the problem is there can be so many different combinations. The first child could have albinism, and the third child can have albinism, and the second, fourth, and fifth could be normal. Okay, or it can be the first 2 that are normal, and then the last 3 have have albinism. Because of the different combinations and their probabilities becoming more difficult, you want to make that task easier to solve by applying the binomial expansion. So to apply the binomial expansion, it takes on this form of this equation, and I want you to pay attention to this, because as we get into Hardy-Weinberg later on in this term, this equation is going to be very, very important. Okay, so the P is going to equal the probability of one event, and Q is going to equal the probability of another event or an alternative event. N is going to represent the number of times the event occurs. Okay, so let's say for this example of the parents having a child with albinism versus having pigmentation or normal, we would say that P would be the probability of the child having albinism, and we can say that P would be equal to 1/4. Okay, equal to 1 out of 4. And then the probability of them having a child with pigmentation would be 3 out of 4. Now, we can say this because we know that albinism is a recessive disease or the recessive disorder. And so you would need to have 2 recessive alleles to produce a child with albinism. So it will be 1 out of 4. Okay. Now, in this case, if you're going to apply the binomial expansion, let me go back. This would be, you would apply the n value here to be 5. Okay. You would apply it to be 5, and then you would actually expand out that binomial. And you can see this in detail on page 59 in the textbook. But it will help you understand how to calculate problems like this when you have more complex situations that cannot be easily applied just by using the multiplication and or the addition rule. Okay, so definitely check out page 59 in the textbook to see this problem full out. Okay, so hopefully this coefficient table right here should bring back memories of Pascal's triangle. Okay, and the number in each row represents the coefficients of each term in the binomial expansion, which we just showed there. So P plus Q superscript N. Note that each number in the triangle, except for the first, is equal to the sum of the two numbers directly above it. Okay. So this should be helpful in understanding or the coefficients in the binomial expansion. Okay. You may also see it using A and B. Okay. So the variables may change. And you can still apply the same concept here. This is prerequisite information that you guys should know. This is just a refresher. There are problems that you can try out, problems on page 60, to apply the concept of binomial expansion. And as we start getting into more problems, you will see that. But I strongly suggest that you go and do your homework problems, because there are homework problems in there to help you with binomial expansion. Okay. All right, so we've been talking about these different crosses and how you can apply mathematical concepts to determine the probability, the likelihood of something happening. In the case of a cross, the inheritance of certain genotypes or alleles being passed on. Now, there is something called a test cross. Okay, you can apply a test cross, and you see this in figure 3.8 that we have already gone over. If you take an unknown genotype with a known genotype, an example would be, an unknown would be a phenotype that, oops, let me go back, sorry. An unknown would be, you have a phenotype that shows a dominant phenotype, but you don't know. I'm going to put this looks so bad, guys. I'm so sorry. But this is what happens when you write with a digital pen. Right? So you have a phenotype that is dominant. Okay, and I'm just going to put D for dominant. But you don't know the genotype. The reason why you don't know is because, remember, the law of dominance, as long as one dominant allele is present, it will exhibit the dominant phenotype. Okay, so you could have this genotype, big T, little t, and produce a tall plant, or you could have big T, big T. You don't know the genotype. The test cross will allow you to determine that by breeding or crossing a dominant unknown genotype, okay, dominant phenotype with an unknown genotype with a known genotype. The only way you'll have a known genotype is if it's recessive. So in this case, it would be lowercase t, lowercase t. So a wrinkled, because that's only, the homozygous recessive is only going to produce one phenotype. Okay. Doing this will give you, or allows you to do, a test cross, because the offspring will tell you what the genotype is of the unknown. How? Go back to the Punnett square and look at it. If we were to set up a Punnett square, okay, we'll see if we can do this somewhat quick, okay, given the time, right? Remember, we know this is known with our unknown. You can then apply the Punnett square, and it will tell you, and in this case here, we're going to do the other one, okay? This would be, again, still with a Testcross, the known, which is little t. Okay. The ratios and the phenotypes are going to be much different. Okay. Here, we're going to have 100% are going to be carriers. So all of them are going to produce tall phenotype, and their genotype is going to be all carriers. In this case here, it's not going to produce 100% of the same phenotype, because here you have these two recessive alleles. So you're going to have some, in this case, right here, both of these are going to be recessive. So you're going to have 50% that are going to be recessive and 50 that are going to be dominant. This would let you know that the unknown was a carrier. Okay. This is how Testcross allow you to see or determine the unknown genotype. Okay. And the ratios in these crosses are going to be very straightforward in a monohybrid cross. Okay. They're very straightforward. You can look at tables 3.5 and 3.6 to help further understand the differences between the genotypic ratios and the phenotypic ratios, and with given examples. I would definitely employ you to do that. Okay, so here's one of the tables for 3.5 that shows you the phenotypic ratio and the corresponding genotypes. And this is table 3.6 that shows you the genotypic ratios for what we would consider a simple genetic cross, okay, for a single locus. So just one specific gene with the two alleles, either dominant or recessive. Now, those monohybrid crosses are much simpler. They're less complex because they're only dealing with one specific gene and just the two alleles. But what happens when you're looking at multiple traits? Okay, that would be considered a dihybrid cross. And this is also something that Mendel worked on, where he began to cross varieties of peas that differed in two characteristics. Okay, and so in that case, he performed a dihybrid cross. You can look at the word di. Okay, the prefix means two. Okay, so he looked at two different characteristics. He crossed one homozygous variety that had seeds that were round and yellow with another homozygous variety that had seeds that were wrinkled and green. Okay. What he found was that the seeds of all the F1 progeny were round and yellow. And then he allowed the F1 to self-fertilize, which you see here. And he obtained the following progeny in the F2. 315, I believe it's 315. Let's see. Yep. 315 round yellow seeds, 101 wrinkled yellow seeds, and 108 round green seeds, 32 wrinkled green seeds. So when you look at those raw numbers, if you look at them based off of ratios, it's a 9:3:3:1 ratio for phenotype. Okay, 9:3:3:1. Now, the dihybrid crosses are going to produce something that's much different than the monohybrid. The monohybrid is producing potentially 4 offspring, so each cell represents a potential offspring or progeny. And the dihybrid cross, it's 16. Okay, it's 16. And that's how you do the same concept of putting the potential alleles there for each potential gamete, but you'll bring them down and bring them over. And so in this case here, okay, at the top, you're going to bring down the big R and the big Y. And in this here, you're going to bring over the big R and big Y into this until you get this specific genotype. Okay. So you do the same thing as you did in the monohybrid. The only difference is you're dealing with more than one trait here. So when working with dihybrid crosses, they reveal the principle of the independent assortment. Okay. What is the independent assortment? The independent assortment states that alleles at different loci separate independently of one another. Okay? So they're going to separate independently of one another. And a common mistake is to think that the principle of segregation and the principle of independent assortment refer to two different processes. Okay? The principle of independent assortment is really an extension of the principle of segregation. The principle of segregation states that the two alleles at a locus separate when gametes are formed. The principle of independent assortment states that when the two alleles separate, their separation is independent of the separation of the alleles. So they're really, one is a branch off or an extension of the other. Okay. And this ties back to, again, meiosis. Okay. It relates back to the process that we talked about in chapter 2 in meiosis. Okay. Gametes are located on different chromosomes and they will sort independently of each other. So if you look at this diagram here, it helps you to understand the principle of independent assortment, but it shows that the results from the independent separation of chromosomes in anaphase 1 of meiosis. So I want you to be able to tie these concepts that we're talking about back to an actual real life application. And this is talking about the processes and the stages of meiosis. So you'll see here that this cell contains 2 pairs of homologous chromosomes. Okay. 1, 2. They undergo replication. And so now you have the homologous chromosomes, okay, that are going to each pair up and independent and separate independently. Keep in mind you have the centromere here, okay. And here you have the different ways in which they can line up or separate. And you'll see that genes located on different pairs of chromosomes assort independently, producing different combinations of alleles in the gamete. So this is another layer of genetic variation. All right, so with these dihybrid crosses, you can apply, again, the probability and the branch diagram to the dihybrid crosses, just like we saw before when we were doing the probability. You can apply the same concept to dihybrid crosses. And you can see here, they're just using the branch diagram, as you see here, may be more helpful. But a branch diagram, it can be used to determine the phenotypes and expected proportions of offsprings from a dihybrid cross, particularly because they tend to be, if you've ever done a dihybrid cross, it is a little bit, takes a little bit more time than doing a monohybrid cross. But you can still apply probability to that. I would strongly employ that you guys work on the problems and the homework, because these homework problems are going to be very, very helpful in your understanding of doing the dihybrid cross and applying the probability. So here you can see here that they're showing the 3/4 round, the 3/4 yellow. If you apply that multiplication rule of 3/4 times 3/4 gives you 9/16. So it's an easier way of applying the probability to the dihybrid cross by just using a branch diagram. Especially if you're going to be taking the MCAT or any tests, this may be an easier way for you to do so. Okay. So with the Dihybrid Testcross, you're going to do the same concept, right? You're going to take a known genotype, which is going to be the recessive, and cross it with an unknown genotype, but a known phenotype. You're going to cross it, and then you can do the probability and the calculations to identify the likelihood of the potential offspring. And so in this case here, you have 1/2 round that comes from up here, okay? In this case, you have 1/2 carrier. Bring it down here. 1/2 times 1/2 gives you 1/4. Okay. Same thing here. 1/2 round, 1/2 green produces 1/4. Okay. Same concept, doing the test cross. So what are these observed ratios of progeny that may deviate from expected ratios by chance? One of the things that you can do to understand or determine if these expected or these observed ratios deviate from what is expected, you can do a chi-square test. Chi-square is another term for that is the goodness of fit test. It's a statistical test that allows you to evaluate the role of a chance in producing deviations from observed and expected values. Okay, this test is going to provide information on how well observed values fit expected values. All right, so in doing this, you can see here, you have to be given the observed. Okay, so in this case here, you have purple and white, and it was observed 105 were purple, 45 were white. You total up the observed column, okay, and then based off of Mendel principles, right? If white is recessive and purple is dominant, then you would expect to see the 3-to-1 ratio. So you would take 3/4, multiply it by 150 because that's the total. Okay, and you get 112.5, and then 1/4 times 150, and you get 37.5. You're then going to take these two values and do a chi-square to determine if what you observed, okay, corresponds or fits with what you expect based off of applying Mendelian principles. Okay, so then that's how you calculate the chi-square, and this the chi-square is given here. I will also give you some help or a cheat sheet, if you want to call it that, to help you understand or remember how to calculate the chi-square test. But there are also practice problems in the homework to help you and throughout the book. So I want to make sure, I strongly suggest that you go through the problems and do this. Okay. Once you calculate your chi-square value, you're then going to calculate the degrees of freedom. And your degrees of freedom can be found by doing n minus 1. Okay. N minus 1. And in this case, it'll be 2, 1, 2, minus 1, which is going to give you a degrees of freedom of 1. And then you have to go to the table, which I'll show you all. And it'll be posted for you in the textbook on how to calculate or determine the significance of your answer or your response. All right. What I want you to do for calculating the goodness fit test, I want you to try problem 38. This is in the textbook. Okay. This is in the textbook, problem 38. And what I love about this book is that it's going to walk you through it step by step. If you look on page 68, you will see that is where the critical values table is located. So if we have our chi-square value and we have our degrees of freedom, we can then use that to determine our level of significance. Okay. I want you to go through again and work on this problem. Go through it step by step. This entire problem is on pages 68. My apologies. It's on page 68. Let me activate this pen again. Okay. And 69. Okay. So I want you to go through this entire problem from start to finish. And the book does it step by step. So there should be no reason why you cannot calculate it. There's also buttons that say hint to help guide you. So if you do get lost, it's there for you. Okay. So we finished chapters 1 through 3, and you should be doing your homework. There's at least 5 problems per each chapter, so it doesn't take a long time for you to go through these problems. They're randomly sorted, but they should help you with practicing in preparation for your exam.